Your last comments, Maja, were particularly important, I think, in terms of understanding the issues associated with the collection of data appropriate. Because at this stage, it's accurate that we don't have what would qualify as big data for populations like the ones you were describing, the autism population being a great example. What are the barriers? And this is really a general question to everyone here for accessing these types of data. You know, obviously what we're doing here is trying to build technology that continually enters the home, enters the private worlds and environments of these individuals across the life course. And that naturally barriers exist. And there are probably two major barriers: sociological barriers and sort of clinical barriers or medically relevant barriers. So I'd love to just open it up for a dialogue on these points. And whomever wants to jump in, please do so.
I'll jump in just to say that I think there's tremendous reluctance on the part of people who do have the technology. So for example, we do have now technologies that are in our home, which are invading our privacy because we're willing to give it, right? So, you know, I have lots of Amazon listening products in my home. I love to command them to do things for me. It's great, right? Those kinds of platforms could be used to collect these kind of data, but those providers, for good reasons, are reluctant to go into that space. We as researchers, on the other hand, cannot go into homes at scale because we don't have access to these hardened products, right? Everything that you heard about, you know, all of us are kind of pushing the boundary of what's possible technologically. It isn't quite market ready, it isn't hardened. And so there's a gap, right? They're completely market ready, deployed products that aren't collecting the data we need, or if they are, then they're not sharing with us in research. Or we have things that are just on the bleeding edge of being ready to be deployed. That's my 2 cents.
Yeah. So I can add my perspective to it. So actually, one of the things that motivated us to work on using wireless signals to capture a lot of physiological signals is 2 things, actually. One thing is adherence and compliance is noted as a very, very big problem. So patients actually don't even take their medications. So we wanted something that does not require any overhead from the patient or from the monitored person, so that it works completely passively. You don't really need to do anything. So this is one thing. The other thing is, of course, cameras are interesting devices, camera and like audio recording devices such as Alexa, for example, but they invade privacy at very high level, and people are not willing to deploy cameras all over the place. So it's kind of like abstracting enough so that you still have useful information, but at the same time, it doesn't show how the person looks and what they are wearing. So I think these are 2 factors that are, of course, important. Also, despite that, I think privacy is still very, very important, and whoever is working in this domain has to be very careful about what they collect, ensuring that the patient knows what is being collected, and ensuring that everything is secured and treated according to the user's interests and desires.
Yeah, very good. Looks like we have a question from the audience.
Yeah, we are a machine learning company, a precision health company interested in continuous modalities, cortisol and glucose. And we're wondering, you mentioned, Ali, that glucose is not informative from sweat. Glucose picked up from sweat is not informative. Have you tried to collect cortisol? And then for Zhenan, how many years between we can get between now and continuous cortisol monitoring through sweat?
Okay, so for the first part of the question, for glucose, this is a work that we have looked at over the last 3 years or so. We have looked at a lot of subjects, healthy, diabetics, you know, fasting, no fasting, different time intervals, all the imaginable studies. So that is really what is needed to see if there are correlations, if there are any strong correlations, no correlations. And what we have seen for glucose, and this is a work that we have already submitted, it's not published yet, but I can, I guess, share the main findings, is that the absolute values, at least, of glucose in sweat and blood are not correlated. And now, that doesn't mean that the dynamic response is not important. That's something else that we are looking at. But the absolute values are not correlated.
Can I ask a follow-up question before Zhenan talks about cortisol? So when you say it's not meaningful enough to dose insulin, or it's not meaningful enough to know for a healthy individual who wants to know how they spike on cheesecake?
The answer is both. And I think the problem is, from blood to sweat, there are two filters. The way to think about it is blood and sweat are correlated through two filtration processes. That's the simplest way to kind of describe how sweat is generated. Now, we do not quite understand whether through these filtration processes there's a time lag there that prevents us from monitoring the proper concentrations that are happening dynamically, the changes that are happening dynamically in blood, or whether the concentration changes are just so small that we cannot measure it given the technologies that we have. We have done a lot of off-body measurements as well. So there we use, of course, the gold standard analytical techniques, and still we do not see any correlations. And this has been a very kind of controversial topic in the last 30 years, last 40 years, maybe even last 50 years. There are papers that say there is a strong correlation. There are papers that say there is no correlation, and something in between. And we spent 3 years looking at it. And to this day, we do not have any, it's based, what we have seen is the correlation is extremely weak.
Wow. So in the case of cortisol sensing, there are point of care cortisol sensing devices, but there hasn't been any continuously monitoring devices. So what we are able to do currently is we're able to show continuous monitoring or detection into the micromolar range pretty reliably. But for concentration in sweat, it's in the order of nanomolar. So we still need to improve the sensitivity by at least 10 times. We're working with materials, new materials that are more sensitive, and also strategies to concentrate samples of sweat continuously. So it's still ongoing work.
Question from the back.
Yeah, so I had a question about the Emerald system. I know that you were saying that it's related on EMF, like electromagnetic frequency, and how it monitors things from the people, like breathing and heart rate. But are there any negative side effects of EMF or electromagnetic frequency? I know that I've read a few articles that talk about how it can cause cancer. Like in our iPhones, in the settings menu, it says that like if you hold it on our ear too long, it'll give you cancer. So like, what are the negative side effects of EMF?
Yeah, so the question about safety of using the electromagnetic waves, and I'm sure, so the first thing is that, of course, if you have very high power, then it becomes, then you might start having issues. But really, the power that we use in our device is about 1,000 times lower power than your Wi-Fi. So I'm sure you have a Wi-Fi at home. I'm sure probably all of you guys and myself, we all have Wi-Fi at home. And if you are not worried about that, then think about something that's like 1,000 times lower power. So, and that actually one of the challenge, how can you get all of these physiological signals and all those measurements from some signal that is really weak?
Thanks.
Yes.
So, Ali, you mentioned about the reliability of the outcome measures and the drift. I was wondering, this is open to everyone, but if you could talk about like how far away are we from having reliable outcome measures that we can use as like endpoints for clinical trials?
Yeah, very, very good question. So I, put a big list of things that we have, the sensors that we have already published or submitted from our group in terms of the sensor technologies, the different analytes. I would say about 10 of them are quite robust today. Making the sensors robust enough is not something that the students love to do. Yeah. It's pure engineering. You cannot get science or nature, but it is fundamentally the most important research topic in this field, in my opinion. If you have sensors that you can report that is detecting X and Y, but not without drift or low enough drift, without the good enough accuracy that you need, I believe the information will not be informative. And so for the ions, for example, the ion sensor selective membranes that we use for those, the chloride, the sodium, potassium, those sensors are pretty good pH. For heavy metals, the sensors are pretty good that we have in our lab. We do have about 10 of them, I would say, that I feel pretty comfortable with the results that we get.
Thank you.
Yes.
Me again? Hi, so I'm Paul. I'm a postdoc at Stanford here, and I study neuromodulators in the brain. So my question's for Professor Bao. I was actually wondering, because there's been a huge challenge in trying to analyze in vivo in the brain levels of neuromodulators, of neurotransmitters, both in space and time. And I was wondering what progress you've made with your materials to measure neuromodulators like dopamine and serotonin in particular.
Yeah, so we have an electrochemical-based sensor that is made of nanomaterial that is sensitive to dopamine and serotonin selectively. So they can be incorporated onto those elastic material and being able to insert into the brain. So we were able to show detection using these kinds of probes with comparable, sometimes even better sensitivity compared to those of carbon fiber, which is what's typically being used in the labs. What we hope to do is to be able to produce these dopamine sensors on not only just one sensor. In the case of carbon fiber, you can only have one sensor, but we can have arrays of these sensors and sensors that can sense dopamine coming from different directions and then give more information that compared to the current probes.
That's really fascinating. Is it like it's a specific biochemical sensor, you said?
Right. So it's an electrochemical sensor. Yes.
Mm-hmm. Okay. Thank you.
Yeah, this topic of making sure that the sensors that we're utilizing in more ubiquitous solutions match with signal specificity and sensitivity. What is gold standard in the field is an important part of the regulatory approval process, right? Moving things to market and overcoming some of the barriers for entry in commercialization, for example, or in widespread adoption and delivery to the bedside or to the homes as an alternative solution. But when those signals that you're measuring and the sensors that you have higher specificity and sensitivity than the gold standard, how do you know? What do you do with that? You're starting to beat what's currently gold standard, what the FDA is asking for example. Have you been thinking about this? Do you think about that regulatory compliance framework? I'd love to hear some thoughts on that.
I can give some information about this because we work with pharmaceutical companies who are particularly interested in new digital biomarkers. So I showed, for example, and one of the tests in Parkinson's, for example, when you approve a drug is the gait speed. But then people want to say, okay, so actually gait speed in the lab, that's the way you approve the drug, but this is not representative because we know that when the patient comes to the doctor, they put a show, and it's just, this is not how they walk at home. The spouse would be saying, no, that's not really how he walks. But now if you measure it in the home and you have not just gait speed, you have also, you are measuring balance, you are measuring even like elevation and like going upstairs, going downstairs. Doctors know that this is more meaningful, but at the same time now you have to go to the FDA and say, okay, so we want to change the metric, or this is a new metric. Mm-hmm. And by being able to measure more and monitor new things, now so one axis is the accuracy. If you are more accurate than the gold standard. Another thing is that if you enable new measurements that are not possible and therefore they were not included in the typical way, what's called endpoints, for example, for drugs, then how do you convince the FDA and you convince them eventually, but the process is so long.
Exactly. Yes. I didn't mean to jump in and take away a question from the audience, but I was excited about this topic.
It's just a very general question. I'm a research scientist, actually work for Mike Snyder. so a very wearables person, and so we actually look at all the different omics. So that'd be interesting for some of the team here. I just have I'm also a consumer, and I can see myself using all these products, and just a general idea of when do you see this coming up to market, and what is really as a scientist who might go into this field, what are the stepping stones or the fallbacks of this type of work?
All right. Well, so let me just speak from the perspective of robotics. So robotics has exploded in the last few years, and pretty much everybody I know in the academic world in robotics has a startup because if you don't, what's wrong with you? So we all do, right? And so that's just a slice of the field. But the same is true for AI, machine learning. And I think the answer to the question about what that means for health in particular is that they're going to be, I think, thin sliced products, and they're already coming out. In some sense, especially in the app space, a lot of things that are not validated at all, and then they just die because they're basically, worthless, but somebody made a little bit of money maybe or not. And a lot of things I'm actually, because of all the regulatory issues with getting data, I think probably what will happen is products will come on the market, people will buy them because they're desperate to, help their child. And that may be how we will get more data, that whether it fuels science or more product development. So I think there will be a graveyard of products, but some data will result in many lessons. And that's pretty much how the market works. It's not really efficient, but there it is. That's my guess. That's what I'm seeing right now. The products are already out there. They're already coming out. So if you look like at Kickstarter, you'll see a lot of these things. The other thing that I'll just throw out, in particular, speaking about this at Stanford, there is a huge reluctance on the part of the VC community, especially Silicon Valley VC community, to invest in anything related to medical stuff. They're just really worried about all kinds of liability and not making enough money issues. Boston is a little bit better, but there's a lot of it's a fact, right? This is why they put me on the panels, because I'll say pretty much anything. But as someone who has done a startup in the health space, I find it incredibly frustrating that the atmosphere in the VC world right here in Silicon Valley is incredibly pessimistic, conservative, unwilling. Entertainment, yay. Health, ooh.
Great.
Resonate.
Do we have any additional comments on this?
Yeah, I resonate your comments on the VCs being very conservative on the medical devices. For the non-invasive blood pressure monitoring product we try to commercialize. This is the second startup I've been working on. The first one was on materials. I thought, materials startups were difficult to raise funding. But the medical one, even though it's not even implantable, just attaching a watch onto the person, but there were just so many concerns about FDA approval, how long it's going to take to the market, and it was very challenging for fundraising.
Exactly.
Yeah, well, I guess I'll comment also on this since I know a thing or two about it. But coming back to this notion of creating solutions that can simultaneously impact things that we care about at the point of care while generating data that represent better endpoints ultimately than what we are trying to match in the regulatory approval process is an entirely pioneering thing. This is the tip of the spear, and you guys are the Avengers. I just saw The End Game. It's a great movie.
It doesn't need more advertising.
But I think these things are going to start to catch on, and I think the venture capital community is going to realize the opportunities that these ideas represent. We have another question back in the audience.
My name is Sarah Eagleman. I'm a postdoc in the anesthesiology department here, and I was just thinking about with Dina's technology, recovery from surgery in patients and monitoring them at home. And I was just curious the limits of your technology. Like, I'm envisioning somebody with a family at home, and you could have 4 people, and then a couple caregivers come in, your housekeeper come in, whatever. What's the ability of your technology to discriminate between the different people, and how well are you able to capture the different physiological measurements that you're talking about?
Yeah, so I mean, the technology is designed for the home, so it would capture multiple people. Like I showed you when the guy was walking, there is a red dot. Now, if there were two people, you see the green dot and the red dot, or the blue and the green and the red. There is a different question also implied in what you mentioned, which is like, okay, so if you get those people, how do you distinguish who's who? And for that, we also, based on the various physiological signals, we build a machine learning classifier so that we can focus on the patient of interest, and we don't monitor the rest of the people in the environment. So it scales to multiple people. Of course, it doesn't scale to this number of people in this room. Is designed more for a home environment.
But it's based on patient condition then? Or like your machine learning algorithm, when you identify that person, is it based on?
A variety of things. So just gait actually has been shown to be really different. Like every person walks differently. So we particularly use the gait of the person to distinguish who's who. And then once we do that, we ignore everyone except for our patient.
Thank you.
I'm not sure which of you two was first.
Okay, I'll go. So, Dina and Maja, I'm an economist from National Institute on Aging, and you guys have influenced me enough to run a robotics program and an assistive technology program. So when I hear that the VCs are not willing to invest, I'll say, look at a National Institute of Health, we are interested, so that's
all right.
Number two, I would like some advice. What can we do better at National Institute of Health or at other federal agencies to invest on what you're working on to make it viable and make sure it goes out in the street within the next 5 years? Because this has a lot of value, right? Whether it's the socially assistive robot or what Dina showed on tracking individuals. Because from gait speed, we can predict many things, just not Parkinson's. We can predict Alzheimer's disease or dementia forming. So again, any insight will be helpful. Thank you.
I have some opinions on this. Thank you for the kind words. In fact, it is the case that in this country, the largest investment comes from NSF and NIH in terms of starting companies. So, VCs love to take the credit, but the reality is if you look at those programs and how much technology is developed and as a spin out. So yes, thank you. But I do have a large caveat. So I have actually served on an NIH panel. I've been, in that world. NIH is extremely conservative. So VCs are conservative about losing money. NIH reviewers, or panelists in particular, sitting panels, are very, very conservative about funding things that are different from what they're used to funding. I don't want to generalize over all institutes. So I know every institute is different in its own way. But sitting panels at NIH, I think, are the death of innovation. So one of the reasons, I'm sorry, but I'm just going to tell you what I think. So I've been involved with NSF. I've been involved with NIH for 20-some years, and I think one of the advantages at NSF of panels constantly being new blood, is that you don't get these dynamics of a group of people who know each other, they know what they want to fund, and they only want to fund that. And so it's hard for NIH to actually be as innovative as it could be. Sorry.
That's fine, I'll take that.
But we love you, and keep doing it.
Actually, let me add one thing. So NIH traditionally does not work with computer scientists and electrical engineers. Now there are so many opportunities that are cross-boundary, cross-disciplinary, bringing computer scientists, electrical engineers to the health domain. And I think NIH needs to figure out a way of incorporating that knowledge and reaching out to those people. If you have your traditional people on your panel, they are not going to fund these things. For a variety of reasons, the basic one is that they just don't understand that kind of technology, so they cannot review it. So, and I've seen NIH in the last year or so, and actually I'm working with some panels at NIH and would love to do more. But it's basically reaching out and trying to understand that gap and trying to figure out ways to remedy it.
Further comments on this topic? Thank you. Next question.
Thank you. I'm a physician and I work in the space of integrative medicine, functional medicine, which basically means I try to bring all the non-pharmaceutical therapies into Western medicine. And I end up taking care of a lot of patients with mystery illnesses where they've been through the whole, system and nobody really knows what's going on, and neither do I, but I kind of try to tease it apart. So as I was listening to the presentation on the wearables and the invisibles, my brain was like kind of thinking about, well, how do we know that these materials don't interact with our physiology, stimulate the immune system, cause inflammatory reactions? Because certainly I do see patients who have sometimes very small implants. I just had another person who took 45 minutes to tell me all of her symptoms, and then we found out she had a screw in her head, and now she's reacting to the metal. And that's just one of her problems, but it's something significant that we have to think about. So is there thought given to the materials that are being used for these invisibles? How do we know they're safe? Because I don't believe in this country we do really good testing to make sure things are safe before they're released out into the public, and that scares me.
Well, certainly for, I can comment on that. Certainly for new materials, we don't have a lot of information on what their impacts are going to be, and this is subject to studies. For implantable devices, we try to use materials that are already known or tested for implantable applications, and then repurpose them or redesign them. But certainly it's still a long road to go in that regard, and we have to keep learning and find out. Yeah, it's probably likely these things are going to be applied first in places where there's no other cure for the disease, and then that's maybe the entry point. But unfortunately, we won't know everything or be able to predict everything when we start to deploy these new technologies.
Sure, and even similarly with EMF radiation, sure, your signal might be much less strong, the strength may be much less strong, however, it's right on the body. So do we have any data on that?
So there are two things. One thing is that, so how do we do studies? I mean, we do studies. I mean, in a sense, you can think about Wi-Fi is really a very huge empirical study that has been, or empirical evidence. We have it probably with like almost 1 billion of people who have it. There are no reported and well-tested evidence that there is an actual health issue. So, I mean, everything, of course, you can't really, this is like a negative result. You can't really prove negative result. But what you can do is to look at the evidence. And I think particularly in evidence for very low power devices like Wi-Fi, I mean, if there are very strong evidence or there is really a strong effect, we would have data by now that is that we can study it effectively, because these devices are everywhere in almost every home. Mm-hmm. So again, I can't give you negative, I cannot prove the negative for you, but the evidences are all in the right direction. Yeah.
I do have patients who claim they can feel Wi-Fi, and I was like, okay, so you can't come to clinic. So we went to a place where I could turn it on and off, and she could kind of tell me whether it was on or off, which really kind of freaked me out. But
well, I think these are great. These are great points. And I think these safety issues continue to be something that we must question and interrogate very carefully, but it should not prevent the pioneering work and the disruptive innovations that these illustrious individuals are doing. And I look forward to more, of course. We have time for one more question.
I'll try to keep it brief. Thank you so much for a wonderful panel. My question is also related to material safety, but more on the environmental side. How much thought and what kind of discussions are gone into the environmental impacts of your devices in the material selection, development, and deployment?
Maybe I can, let me see if I can answer that question. Actually, linking back to the previous question, this issue of safety and impact on environment is going to be very important. Many of these devices are going to be disposable. And there have been some studies regarding looking at different materials and their health and safety impact. In the case of health impact, lotion, your sunscreen, they are not FDA approved. But believe it or not, what we have seen at least is that cosmetics, a lot of these impurities that you put on your body actually get reabsorbed. They get absorbed through the skin. And it's just mind-boggling that it's not even discussed. And so I mentioned briefly in my talk that, for example, we ask our patients for some of the glucose studies not to use certain shampoos or soaps. We actually went as far as looking at the composition, as far as we could tell at least. And many soaps and shampoos, for example, have sugars in them. If you're trying to study glucose, right, and what you put on your body already has sugar, what are you measuring? Now, it is really important to think about using materials that are biocompatible, that are safe. Devices, in terms of wearables, my vision is that the active elements should not be in contact with the skin. So our newer generation devices, in fact, the active elements are actually separated from the skin. We have a microfluidic, the microfluidic inlet is in contact with the skin. But these are very important questions moving forward. Yeah.
Do we have any other comments? We, I think, stand between you and lunch, so let me take this opportunity to thank the speakers one more time. It's an incredible panel. I hope you enjoyed it. Thank you very much.